Chapter 13. Photosynthesis in higher plants. 1. Photosynthesis is a dash process. Physiochemical. 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 2. Co-absorb dash gas. CO2. 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 3. All living forms on Earth are ultimately dependent on dash for energy. Sunlight. 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 4. Dash is the primary source of all food on Earth. Photosynthesis. 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 5. Who showed that chlorophyll is located in special bodies called chloroplasts? Julius von Sachs. Julius von Sachs. Julius von Sachs. 6. Dash illuminated a green alga dash placed in a suspension of anaerobic bacteria, TW. Engelmann, Cladophora, aerobic, TW. Engelmann, Cladophora, aerobic, TW. Engelmann, Cladophora, aerobic. 7. Dash proved that the O2 evolved by the green plant comes from H20, not from CO2. Cornelius van Niel. Cornelius van Niel. Cornelius van Niel. 8. Bacteria accumulated mainly in the region of dash and dash light of the split spectrum. Red and blue. Red and blue. Red and blue. 9. Who showed that sunlight is essential for photosynthesis? Jan Ingenhouse. Jan Ingenhouse. Jan Ingenhouse. 10. Jan Ingenhouse did an experiment on an aquatic plant. True. 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 11. Who showed that it is only the green part of the plants that could release oxygen? Jan Ingenhouse. Jan Ingenhouse. Jan Ingenhouse. 12. Dash provided evidence for production of glucose when plants grow. Julius von Sachs. Julius von Sachs. Julius von Sachs. 13. Glucose is usually stored as dash. This was told by Julius von Sachs. Julius von Sachs. Julius von Sachs. 14. Dash performed a series of experiments revealing the essential role of air in growth of green plants. Joseph Priestley. Joseph Priestley. Joseph Priestley. 15. Who showed that green parts of plant make glucose? Julius von Sachs. Julius von Sachs. Julius von Sachs. 16. Priestley discovered dash in year dash. Oxygen, 1774. Oxygen, 1774. Oxygen, 1774. 17. Bacteria was used to detect sites of O2 evolution, sites of O2 evolution, sites of O2 evolution. 18. Priestley used dash plant. Mint. 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 19. The first action spectrum of sunlight was described by T. W. Engelmann, T. W. Engelmann, T. W. Engelmann. 20. The first action spectrum resembles roughly the absorption spectrum of chlorophyll dash and dash. R&B. 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 21. A microbiologist, dash, based on his study on dash and bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction in which CO2 is reduced to carbohydrates. Cornelius van Niel, purple and green bacteria. Cornelius van Niel, purple and green bacteria. Cornelius van Niel, purple and green bacteria. 22. Beer-jar experiment was done by Priestley. 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 Photosynthesis proper. 23. Dash cells in leaves have large number of chloroplast. Mesophyll. 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 24. Chloroplasts align their flat surfaces parallel, perpendicular to the wall when given low light intensity. Parallel, along the wall. Parallel, along the wall, parallel, along the wall. Parallel, along the wall. 25. The matrix of chloroplast is called stroma. 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 26. There is a clear division of labor within the chloroplast. True. 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 27. The membrane system is responsible for dash and dash. Trapping light energy, synthesis of ATP and NADPH. Trapping light energy, synthesis of ATP and NADPH. Trapping light energy, synthesis of ATP and NADPH. 28. In stroma, light-dark reactions occur. Dark, dark, dark. 29. Enzymatic reactions occur in stroma. True, true, true. 30. Dark reactions are light-independent. True, true, true. 31. Dark reactions occur in darkness only. False, false, false. 32. Identify the organelle shown in diagram. 1. Label A, B and C. Diagram. 1. Chloroplast, A. Lipid droplet, B. Starch granule, C. Ribosome. Diagram. 1. Chloroplast, A. Lipid droplet, B. Starch granule, C. Ribosome. Diagram. 1. Chloroplast, A. Lipid droplet, B. Starch granule, C. Ribosome. 33. Diagram. 2. Shows the absorption spectrum of chlorophyll A, B and the carotenoids. Identify the curves A, B and C of the pigments. Diagram. 2. A. CHLB. B. Carotenoids. C. CHLA. Diagram. 2. A. CHLB. B. Carotenoids. C. CHLA. Diagram. 2. A. CHLB. B. Carotenoids. C. CHLA. 34. Name the 4 pigments found in leaves. Chlorophylla, bright or blue-green, chlorophyll B, yellow-green, xanthophylls, yellow, and carotenoids, yellow to yellow-orange. Chlorophylla, bright or blue-green, chlorophyll B, yellow-green, xanthophylls, yellow, and carotenoids, yellow to yellow-orange. Chlorophylla, bright or blue-green, chlorophyll B, yellow-green, xanthophylls, yellow, and carotenoids, yellow to yellow-orange. 35. Chlorophylla color is bright or blue-green, bright or blue-green, bright or blue-green. 36. Chlorophyll B color is yellow-green, yellow-green, yellow-green. 37. Yellow to yellow-orange color is of carotenoids, carotenoids, carotenoids. 38. Xanthophylls color is yellow, yellow, yellow. 39. What does X and Y axis represent in the graph shown in diagram? 3. Diagram. 3. X-axis, rate of photosynthesis, Y-axis, wavelength of light. Diagram. 3. X-axis, rate of photosynthesis, Y-axis, wavelength of light. Diagram. 3. X-axis, rate of photosynthesis, Y-axis, wavelength of light. 40. Identify A and B in diagram. 4. Diagram. 4. A. Rate of photosynthesis, B. Absorption. Diagram. 4. A. Rate of photosynthesis, B. Absorption. Diagram. 4. A. Rate of photosynthesis, B. Absorption. 41. Wavelengths at which there is maximum absorption by chlorophyll dash also show higher rate of photosynthesis. A. 42. Dash is the chief pigment associated with photosynthesis. CHLA. 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 43. Most photosynthesis take place in dash and dash color region of the spectrum. Blue, red, blue, red, blue, red. 44. Accessory pigments X. CHLB, xanthophylls, carotenoids. CHLB, xanthophylls, carotenoids. CHLB, xanthophylls, carotenoids. 45. Accessory pigment function. 1. Enable a wider range of wavelength of incoming light to be utilized. 2. Protect chlorophyll from photooxidation. 1. Enable a wider range of wavelength of incoming light to be utilized. 2. Protect chlorophyll from photooxidation. 1. Enable a wider range of wavelength of incoming light to be utilized. 2. Protect chlorophyll from photooxidation. 46. Light reaction is also called dash reaction. Photochemical. 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 47. Dark reaction is also called dash reaction. Carbon. 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 48. What 4 things happen in light reaction? I. Light absorption. 2. Water splitting. 3. Oxygen release. IV. Formation of high energy chemical intermediates i.e. ATP and NADPH. I. Light absorption. 2. Water splitting. 3. Oxygen release. IV. Formation of high energy chemical intermediates i.e. ATP and NADPH. I. Light absorption. 2. Water splitting. 3. Oxygen release. IV. Formation of high energy chemical intermediates i.e. ATP and NADPH. 49. LHC is made up of 100 of pigment bound to dash. Proteins. 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 50. LHC also called antennae. 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 51. In psi, absorption peak is at dash nanometer, hence called dash. 700 nanometers, P700. Dash. 700 nanometers, P700. Dash. 700 nanometers, P700. 52. In PSLL, absorption peak is at dash nanometer, hence called dash. 680 nanometers, P680. Dash. 680 nanometers, P680. Dash. 680 nanometers, P680. 53. Photosystems are named according to the sequence of discovery, function. Discovery. 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 54. CHL, A plus LHC equals PS. True. 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 Electron transport and CHEMIOS MOSIS. 55. Light reaction follows dash scheme. Z. 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 56. The splitting of water is associated with psi, PSII. 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 57. PCL is located in the inner, outer side of the membrane of thylakoid. Inner. 58. Protons and O2 formed is released in the lumen of thylakoids. True. 59. ATP synthesis by cells is named phosphorylation, phosphorylation, phosphorylation. 60. Cyclic phosphorylation occurs in the dash component of chloroplast. Stroma lamellae, stroma lamellae, stroma lamellae. 61. I give light at a wavelength of 690 nanometers to a plant. Which phenomenon will be seen? Which PS will be functional here? Red drop effect, PSI will be functional. Red drop effect, PSI will be functional. Red drop effect, PSI will be functional. 62. Both ATP and NADPH are synthesized in cyclic, non-cyclic photophosphorylation. Non-cyclic, non-cyclic, non-cyclic. 63. Stroma lamellae lack which enzyme and photosystem? NADP reductasen and PSII. NADP reductasen and PSII. NADP reductasen and PSII. 64. In photosynthesis, proton accumulate in lumen of thylakoids, lumen of thylakoids, lumen of thylakoids. 65. In respiration, proton accumulate in intermembrane space of mitochondria, intermembrane space of mitochondria, intermembrane space of mitochondria. 66. Draw the sequence of flow of electrons across thylakoid membrane during in non-cyclic photophosphorylation. Z scheme of light reaction. Z scheme of light reaction. Z scheme of light reaction. 67. The primary acceptor of an electron i.e. dash is located towards the inner, outer side of the membrane and transfers its electron to an electron carrier, H carrier. Plastoquinone, outer, H carrier. Plastoquinone, outer, H carrier. Plastoquinone, outer, H carrier. 68. Relative concentration of protons in stroma and lumen is decreased by which three mechanisms? Chemiosmotic hypothesis, diagram. 1. Protons that are produced by the splitting of water on the inner side of the membrane accumulate within the lumen of the thylakoids. 2. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an H carrier. 3. Reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Chemiosmotic hypothesis, diagram. 1. Protons that are produced by the splitting of water on the inner side of the membrane accumulate within the lumen of the thylakoids. 2. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an H carrier. 3. Reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Chemiosmotic hypothesis, diagram. 1. Protons that are produced by the splitting of water on the inner side of the membrane accumulate within the lumen of the thylakoids. 2. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an H carrier. 3. Reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. 69. CF1 is embedded in the thylakoid membrane. False. 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 70. Dash protrudes out of the thylakoid membrane. CF1. 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 71. The breakdown of the gradient provides enough energy to cause a change in the CF1 particle of dash, conformational, at pace, conformational, at pace, conformational, at pace. 72. At pace and ATP synthase are same thing. True. 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 Calvin cycle, C4 pathway and photorespiration. 73. The products of light reaction are O2 ATP, NADPH, O2 ATP, NADPH, O2 ATP, NADPH. 74. How does O2 comes out of chloroplast? By diffusion. By diffusion. By diffusion. 75. The primary acceptor molecule of CO2 is a 2-carbon compound making a 3-carbon compound. False. Primary acceptor molecule is a 5-carbon sugar ribulose 1,5-bisphosphate, REB-P. False. Primary acceptor molecule is a 5-carbon sugar ribulose 1,5-bisphosphate, REB-P. False. Primary acceptor molecule is a 5-carbon sugar ribulose 1,5-bisphosphate, REB-P. 76. Kelvin used which radioisotope of C for studying algal photosynthesis? 14C. 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 77. First product of C4 pathway is OAA, oxaloacetic acid. OAA, oxaloacetic acid. OAA, oxaloacetic acid. 78. First product of C3 pathway is PGA, 3-phosphoglyceric acid. PGA, 3-phosphoglyceric acid. PGA, 3-phosphoglyceric acid. 79. The name of acceptor molecule in C3 and C4 respectively are dash. REP, PEP, REP, PEP, REP, PEP. 80. REP is a ketose, aldose sugar. Ketose, ketose, ketose. 81. Calvin cycle occurs in all photosynthetic plants. True, true, true. 82. The three stages of Calvin cycle are carboxylation, reduction, regeneration. Carboxylation, reduction, regeneration. Carboxylation, reduction, regeneration. 83. Dash is the most crucial step of the Calvin cycle. Carboxylation, carboxylation, carboxylation. 84. Enzyme used in carboxylation is called Rubisco, Rubisco, Rubisco. 85. For 5 CO2 molecules, how many ATP and NADPH will be needed to fix it? 15 ATP, 10 NADPH. 15 ATP, 10 NADPH. 15 ATP, 10 NADPH. 86. How many turns of cycle is required for formation of 7 glucose molecules? 7x6 equals 42. Dash. 7x6 equals 42. Dash. 7x6 equals 42. 87. Dash is crucial for the cycle to continue uninterrupted. Regeneration of REP. Regeneration of REP. Regeneration of REP. 88. Regeneration steps require no dash of ATP for phosphorylation to form REP. 1. Dash. 1. Dash. 1. 89. C4 pathway is found in plants adapted to dash regions. Dry tropical. Dry tropical. Dry tropical. 90. C3 pathway is the main biosynthetic pathway of CA plants. True. True, true. 91. Calvin cycle is also called C3 pathway, C3 pathway, C3 pathway. 92. Dash anatomy of leaves is present in CA pathway. Crans, crans, crans. 93. In C4 plants dash cells are present around vascular bundles. Bundle sheath cells, bundle sheath cells, bundle sheath cells. 94. The bundle sheath forms only one layer around the vascular bundle. False, false, false. 95. Features of bundle sheath cells. 3. I. Large number of chloroplast. E. Thick walls impervious to gaseous exchange. E. No intercellular spaces. I. Large number of chloroplast. E. Thick walls impervious to gaseous exchange. E. No intercellular spaces. I. Large number of chloroplast. E. Thick walls impervious to gaseous exchange. E. No intercellular spaces. 96. X of C4 plant. 2. Maize and sorghum. Maize and sorghum. Maize and sorghum. 97. Hatch and slack pathway is a non-cyclic process. False, false, false. 98. Hatch and slack pathway operate in mesophil, bundle sheath cells. Mesophil, mesophil, mesophil. 99. In C4 plant, primary CO2 acceptor is dash. It is present in bundle sheath, mesophil cells and form dash. PEP, mesophil, oxaloacetic acid. PEP, mesophil, oxaloacetic acid. PEP, mesophil, oxaloacetic acid. 100. Mesophil cells lack Rubisco enzyme in C4 plant. True, true, true. 101. Analog to Rubisco in C4 is pepcase, pep carboxylase, pepcase, pep carboxylase, pepcase, pep carboxylase. 102. After OEA formation, OEA is converted into dash or dash in the mesophil, bundle sheath cell. Malic acid or aspartic acid, mesophil cell. Malic acid or aspartic acid, mesophil cell. Malic acid or aspartic acid, mesophil cell. 103. In a bundle sheath cell, the acids are broken down into dash carbon molecule. 3. Dash. 3. Dash. 3. 104. PEP is regenerated in which cell? Mesophil cell. Mesophil cell. Mesophil cell. 105. Bundle sheath is rich in dash enzyme and lacks Rubisco, pepcase. Rubisco, pepcase, Rubisco, pepcase. Rubisco, pepcase. 106. In C4 the Calvin pathway occurs in cells. Bundle sheath. Bundle sheath. Bundle sheath. 107. Rubisco has a much greater affinity for O2 than Colorado 2. False. 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 108. REP bind with O2 to form phosphoglycerate and phosphoglycolate. Phosphoglycerate and phosphoglycolate. Phosphoglycerate and phosphoglycolate. 109. Utilization of ATP happens in photorespiration. True. 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 110. No CO2 release in photorespiration. False. 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 111. The biological function of photorespiration is not known. False. 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 112. At low CO2 concentrations, photorespiration will increase. True. 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 113. In photorespiratory pathway there is no synthesis of ATP or NAF. True, true, true. 114. In C4 plants photorespiration does not occur. True, true, true. 115. C4 plants show tolerance to higher temperatures. True, true, true. 116. C4 plants show less response to high light intensities than C3. False, more response. False, more response. False, more response. 117. C4 plant have lesser productivity of biomass. False, greater productivity. False, greater productivity. False, greater productivity. 118. Which three organelles are involved in photorespiration? Peroxisomes, mitochondria and chloroplast. Peroxisomes, mitochondria and chloroplast. Peroxisomes, mitochondria and chloroplast. 119. Internal factors that affect photosynthesis are I. Number, size, age and orientation of leaves, mesophyll cells and chloroplasts. E. Internal CO2 concentration. E. Amount of chlorophyll. I. Number, size, age and orientation of leaves, mesophyll cells and chloroplasts. E. Internal CO2 concentration. E. Amount of chlorophyll. I. Number, size, age and orientation of leaves, mesophyll cells and chloroplasts. E. Internal CO2 concentration. E. Amount of chlorophyll. 120. External factors that affect photosynthesis are sunlight, temperature, CO2 concentration in water, sunlight, temperature, CO2 concentration in water, sunlight, temperature, CO2 concentration in water. 121. Blackman gave his law in the year 1905. 1905. 1905. 122. Draw the graph between light intensity and rate of photosynthesis. 123. Light is a frequent limiting factor in nature. False, really. False, really. False, really. 124. Light saturation occurs at dash of full sunlight. 10. 10. 10. 125. There is a linear, parabolic, hyperbolic relation between incident light and CO2 fixation rates at low light intensities. Linear. 126. Increase in incident light can cause decrease in photosynthesis. True, due to breakdown of chlorophyll. True, due to breakdown of chlorophyll. True, due to breakdown of chlorophyll. 127. Dash is a major limiting factor for photosynthesis. CO2. 128. CO2 concentration in the atmosphere is between dash and dash. 0.03 minus 0.04. Dash. 0.03 minus 0.04. Dash. 0.03 minus 0.04. 129. Increase in conc. Up to dash 7 cause increase in CO2 fixation, beyond which it becomes damaging. 0.05. Dash. 0.05. Dash. 0.05. 130. C3 plants show saturation only beyond dash CO2 conc. 450 microliter per liter, trinc 9x5 to 45. Dash. 450 microliter per liter, trinc 9x5 to 45. Dash. 450 microliter per liter, trinc 9x5 to 45. 131. C4 plants show saturation at dash CO2 conc. 360 mu IL, trinc 9x4 to 36. 360 mu IL, trinc 9x4 to 36. 360 mu IL, trinc 9x4 to 360 mu IL, trinc 9x4 to 36. 132. Examples of some greenhouse crops 2. Tomato and bell pepper. Tomato and bell pepper. Tomato and bell pepper. 133. Light reactions are temperature sensitive. True. 134. Dark reactions are temperature controlled because dash. They are enzymatic. 135. C3 plants have greater optimum temperature. False. C4 have greater optimum temp. False. C4 have greater optimum temp. False. C4 have greater optimum temp. 136. Temperate plants have higher optimum temperature than tropical plants. False. 137. Dash and dash hormones increase photosynthesis. Cytokinin and gibberellin. 138. Dash and dash hormone decreases photosynthesis. Abscisic acid. Abscisic acid. 139. Water has a direct, indirect effect on photosynthesis. A direct, indirect effect on photosynthesis. Indirect, indirect, indirect. 140. Water stress reduces dash availability. CO2, CO2, CO2. 141. It reduces CO2 availability because I cause closure of stomata, E makes leaves wilt, I cause closure of stomata, E makes leaves wilt, I cause closure of stomata, E makes leaves wilt. 142. Chemiosmosis requires membrane, proton pump, proton gradient and membrane, proton pump, proton gradient and membrane, proton pump, proton gradient and 143. When H2S is used in photosynthesis, S2 gets oxidized to S or S042, S or S042, S or S042. 144. Maximum absorption of light is done in dash wavelength range by dash chlorophyll. 400 to 500, chlorophyll B. Dash. 400 to 500, chlorophyll B. Dash. 400 to 500, chlorophyll B. 145. Leaf pigments are separated by dash technique, are separated by dash technique. Paper chromatography, paper chromatography, paper chromatography. 146. What is reaction center? Single chlorophyll molecule, single chlorophyll molecule, single chlorophyll molecule. 147. LHC contains chlorophyll A. False, false, false. 148. Plastocyanin is present on the outer, inner side. Inner, inner, inner. 149. Calvin discovered dash's first stable product of photosynthesis. 3-phosphoglyceric acid, PGA. Dash. 3-phosphoglyceric acid, PGA. Dash. 3-phosphoglyceric acid, PGA. 150. Acceptor molecules have greater, lesser redox potential. Greater, 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 greater.